I'm going to present the topic next generation sequencing. Introduction. So next generation sequencing or NGS is a massively parallel sequencing technique that offers high reading, scalability, and speed, which can be used to determine nucleotides in the genome or targeted regions of DNA or RNA. So simply, it means that there is sequencing in continuous sequencing occurring simultaneously. So the speed is increased. So, what is the need of NGS? So, as Preetham mentioned, Sanger sequencing outputs almost 1 million bases per day, whereas Josh mentioned that human genome size is 6 billion bases, so it will take almost 100 years to complete the human genome for a single machine using Sanger sequencing, which is not feasible. So, there was a need for new sequencing technique that can read the data much faster and correctly. So this is where NGS coming. So for example, NGS from Illumina, the latest machine, the NovaSeq machine can read up to 13 billion bases with 300 to 700 bases per read, and it can also sequence the human genome in 48 hours for around $1,000, which was, as just mentioned, was 3 billion earlier. So the price as well as time was greatly reduced. So almost all of the NGS have can be divided into two basic steps. There's sequencing by synthesis or sequencing by degradation. But sequencing by degradation is not used by many companies because the reading frame, because the number of bases it can sequence is very less, as we don't have big enough gel or enough sequence all and the workflow. So almost every sequence has a given workflow, that first DNA fragmentation, second library preparation, third sequencing, and last data analysis. So we will now look into each of these with the context of Illumina sequencing, as Illumina sequencing almost holds around 90% of sequencing market. So the first step is preparing samples. So these steps include getting DNA, or if we have RNA, we can convert RNA into DNA using reverse transcriptase. So we got the sample. Now the second step is sequencing library. So in this step, we add an adapter sequence. So adapter sequence consists of a capture sequence and a primer. We will see what is the use of capture sequence and primer later. So this image shows the flow cell of Illumina sequencing. So in flow cell, these green and pink oligonucleotides are the capture sequence. So this DNA sequence has this capture sequence. So when the capture sequence bind with these oligonucleotides in the flow cell. So we get a single stranded DNA. Now there's second capture sequence binds to the next capture sequence in the flow cell. So a bridge-like shape is formed. Now DNA polymerase binds to the primer site and begins replication. So a complementary strand is made. Now we denature the DNA and we get two single-stranded DNA strands. So we have a forward strand and a reverse strand. So this process is continued over and over, and we get multiple copies of forward and reverse strand. So after getting all these copies, we selectively remove forward or reverse strand, any of those, and now we have all identical fragments in the flow cell. So after having all identical fragments, we add a sequencing primer to the open end. As Preetham mentioned, in Sanger sequencing, there was a fluorescent reversible chemistry. So the difference in Illumina sequencing is that there is a reversible chemistry. So there is DDNTPs, but instead of a terminator, it can be reversed. So instead of H, there is O allylic O allylic group. So after adding palladium, we can replenish the OH. So this image shows that during sequencing, when this for example, A base is added, we get a color and we took the image, and then after adding palladium, we can now have open OH, and again we can start adding bases. So this process can be continued over and over. This image shows different sequences at different time intervals having different images. So let's take an example of this first. So at first it shows yellow color, then blue, then red, and green. So we can find that the sequence is A, G, C, C, T, and similarly for the bottom one. So that was about Illumina sequencing, and Illumina sequencing was a short read sequence. That is, it have almost 300 to 700 base pair in a single read. And now we have Oxford Nanopore. So Oxford Nanopore is a long read sequencer, and it can read up to 2 megabases. So in Oxford Nanopore, there is a lipid layer and there is a pore. So the lipid layer prevents the current from going from negative to positive to negative to positive. Sorry, and in this pore, DNA or RNA can also go down. So when there is a base incorporated, we see a sudden change in current. So this, for example, when T is given, there is this change in current. When A is this, there is this change in current. So we can measure the change in current and can tell the basis. But this Oxford nanopore currently has a very high rate because in the spore at a given time, almost 6 bases are present. So, and there are total 4 types of bases. So 4096 combinations can be made. So there is a high error rate. And now ion torrent sequencing and sequencing by ligation. So previously we have seen sequencing that is amplified by bridge chain analysis. Both ion sequence and sequencing by ligation is emulsion bead based. So in ion torrent sequencing, we check when each base is added, we get different change in pH. So by measuring the change in pH, we can tell which base is added. And in sequencing by ligation, for example, we only add one base at a time in normal sequencing, but in sequencing by ligation, we thought that instead of one, we can add two bases. So, and there are total of four bases, so we can have a total of 8 combinations. So this speeds up the process of sequencing and reduces time. Also, one advantage of ion torrent sequencing that it measures the pH, so it does not require labeled nucleotides and optics to identify the light given out. So it is cheaper. So that was all. Thank you.